Myself, Yuvraj Sonone, Head and Associate Professor in Botany, Mahatma Gandhi Vidya Mandirs, Samashri Prashant Dada Hire, Arts, Science and Commerce College Nampur, Taluka Baglan, District Nashik. Today, I am going to teach a topic, structure of Tetrasporangiate Anther. Microsporangium. Stamen is the male reproductive organ of the flower. Typically, it consists of two more or less distinct parts, a lower sterile part, the filament, and an upper fertile part, the anther. The sterile median part of the anther is known as connective. Each lobe of the anther bears two chambers which produce microspores or pollens. Hence, it is known as microsporangium. Microsporangium is also known as pollen sac. Microsporangium possesses diploid sporogenous tissue which produces a number of haploid pollen grains. After meiosis, at maturity, anther lobes remain filled with a powdery mass of pollen grains. Structure of Tetrasporangiate anther. Anther has two anther lobes. A cross section of the anther shows two chambers in each lobe. Hence, anther is known as Tetrasporangiate anther. The anther wall. The mature anther wall is made up of the following four layers: epidermis, endothecium, middle layers, and tapetum. Epidermis. It is the outermost layer composed of a single layer of stretched and flattened lignified or cutinized cells. It is protective in function. Endothecium. It is fibrous layer of radially elongated cells. Normally it is single layered but may become multi-layered in some plants. Cells of the endothecium around the junction of the two sporangia are without fibrous thickenings. With the maturation of the anther, a tearing force is caused by the loss of water from the endothelial cells on the stomium, and the anther deices in this region. Middle layers. 1 to 3 layers lying next to endothecium are the middle layers. The cells of these layers are ephemeral and degenerate almost completely before the pollen mother cells undergo meiosis. Sometimes these layers degenerate soon after meiosis, as in Melothria medarspatna, Momordica charantia, etc. Occasionally, one or more middle layers may persist for indefinite periods of time, as in Coccinia indica, Ranunculus, Holoptelia, and Lilium. The middle layers may store food material in some taxa, which is utilized by pollen mother cells. Tapetum. It is the innermost layer of anther wall and attains its maximum development at the tetrad stage of microsporogenesis. It is completely surrounds the sporogenous tissue. Tapetum is of great physiological importance because all the food material to the sporogenous tissue must pass through it. Generally, tapetum is of single layer of much enlarged cells characterized by presence of dense cytoplasm and prominent nuclei. It may be multinucleated or polyploid. Cells of the tapetal layer are secretory or amoeboid in nature with many nuclei, or a single polyploid nucleus with many nucleoli. Types of tapetum. The following two types of tapetum have been recognized in angiosperms. Number 1, amoeboid or invasive or periplasmodial tapetum. In this type, the radial and inner walls of the tapetal cells break down early due to the action of hydrolytic enzymes. The protoplast of these cells enters into the anther locule and fuses to form a common mass called tapetal periplasmodium. This mass surrounds the pollen grains till they are mature and provides them proper nutrition. When pollen grains mature, the periplasmodium also contributes to their exine by providing sporopollenin precursors. Electron microscopic studies have shown that periplasmodium is a highly organized functional structure. The nuclei present in the periplasmodium also divide. It is a primitive type of tapetum found in Alisma, Typha, Tadescantia, Sagittaria, Putamogeton, Butamus, Helianthus, etc. Second, glandular or parietal or secretory tapetum. This type of tapetum is more common in most of the angiosperms. The cells of secretory tapetum remain attached to the middle layer till the development of pollen grains. At the pre-meiotic stage of the pollen mother cells, tapetal cells are very thin and possesses almost all cell organelles like mitochondria, plastids, dictyosomes, etc. Some spherical structure called proubis bodies pass into the anther locule from the tapetal cells, and these are now called ubis bodies. They perhaps help in the formation of exine of pollen grains. The secretory type of a tapetum is more common among angiosperms, for example, Mirabilis zalepa, hibiscus and ocimum. Functions of tapetum. The tapetum is a specialized layer of nutritive cells found within the anther of flowering plants. It is the innermost layer of the wall of the anther. Tapetum is important for the nutrition and development of pollen grains as well as a source of precursors for the pollen coat. It contains dense protoplasm and plays an important role in providing nutrition to sporogenous tissue and microspores besides contributing to the formation of sporopollenin. It plays an important role in the formation of pollen wall. Many specific proteins of the pollen wall are secreted by tapetum. Amoeboid type of tapetum produces callose enzyme necessary for dissolving callose walls of the microspore tetrads and thus helps in their separation into individual pollen grains. Callose enzyme also play an important role in making the pollen grains fertile or sterile. It produces a oily coating over some pollen grains called pollen kits. It helps an insect attractant during pollination and protects pollen from UV radiations. Sporogenous tissue. Microspores or pollen grains develop from the sporogenous tissue. The primary sporogenous cell may directly function as microspore mother cell. Each microspore mother cell undergoes meiosis and form 4 haploid microspores. The process of formation of microspores from sporogenous tissue is known as microsporogenesis. All sporogenous cells of an anther are capable of forming microspores, but usually some of these cells degenerate and provide nourishment to the active sporogenous cells. In some Gentanaceae, the tapetum is not well developed and then the nutritive function is taken up by sporogenous tissue. References. Thank you for watching video. Please like, comment, share and subscribe the channel.